Well, good morning, everybody, and thank you, Yael, and the organizers of the Swedish conference for inviting me. I think, kind of off topic, since I have nothing to do with autism or children, but I'm going to try my best and link up with homeopathy, really, and I think I'm trying to also tease out what will be interesting for you in terms of where we're going with the physics of homeopathy. So, I will just dive in. So, really, I'm going to change a bit the topic from what you have in the program, and I'm going to tell you about really where we're at about the basic research in homeopathy and how we're thinking about building the evidence in homeopathy basic research. And really, why basic research? Well, the problem we have right now in homeopathy is that we don't really have much of evidence about how it works on the kind of physical level. And why we're interested in like how does it work, how does a pill work, is really also because in the long term we really want to show that homeopathy as a whole works, not only as we can do the clinical trials and we have the evidence from the cases, and I'm sure every day you see kind of great stuff happen in your clinics, but how does it work in the pill and or the LM or whatever you're using? And then beyond that, the long-term goal, as we were talking yesterday with Yael, is behind homeopathy is a whole different way of looking at health. And I think one of the reasons the whole kind of construct, mental construct that we have with homeopathy is different is also because of the way the homeopathy works. And I think I'm trying to show in some slides here how fundamentally different and how actually fundamental homeopathy works for health. So let's go in. So I want to show you some of the problematics we have today in research and how we're trying to tackle them. And then I want to show you two pieces of work. One is an ongoing work we have looking at the evidence that we've already have from physical chemistry. And I don't know if you've seen this work by Stephen Cartwright. We've been Facebooking it, tweeting it, some basic results in physical chemistry. And then a brief overview of what we do at the HRI. So I spent a lot of time just thinking, okay, how do I convince the scientific community that homeopathy, there's really something there. It's not just placebo, it's not just water, there's something. So the problems we have is, we need good experiments, and I mean by that they can be repeated. And in homeopathy, traditionally, we've had a lot of problems repeating experiments. That is, one team does it, and then the next team does it, but somehow it doesn't work. So we've had a lot of trouble with this type of problem. It needs to be convincing. It needs to be somehow simple to do. If you have a very, very complex experiment and then you have a result, it's not so convincing as if you have quite a simple experiment and you see something. And again, in the same vein, it needs something like you can do, and you get a result, and it's not too hard to see, what's actually happening. Again, simplicity is going to be a key in convincing people. And then beyond that, we've really got a problem with the theory, because if we don't have the start of a theory about how these things work, it's very hard to talk to scientists. They just go, well, it's just water, guys. Just get real. It's just water. But so we need to find a way to communicate to them. Actually, we have theories about how this works, and yes, it's mostly water, but there's stuff there, and that's actually what does the work. But we need to construct theories so that we can start to communicate. And this theory needs to be solid enough that we can make some predictions. And ultimately, we need to communicate these results, these findings, this way of thinking to the general community of scientists and medical doctors in the most, forceful, convincing ways. So, the theory. So I'm going to go first on the theory because I want to set up what is our current thinking about the theory, and apologize for those of you who were there 2 years ago. How many saw my talk 2 years ago? Most people, okay, not too many. Okay, so good that I can, and the Norwegian people, I'm sorry because you saw this 2 weeks ago. But I want to put the background of the theory of what we're thinking is happening, and then you'll see how this fits in with the experiments that we're looking at, and the general. Everything kind of fits in once you've seen the picture of the theory. So it all starts with quantum theory. Classical theories of water fail at describing homeopathy, but quantum theories can. And we start with the theory of quantum electrodynamics, which is basically the theory of light interacting with matter. And we kind of extend that to look at dipoles in water. Water are polar, and they have little charges which make them a dipole. And we can look at how electromagnetic radiation interacts with water. So this is kind of the basic quantum theory of that. And then the predictions from the two Italian guys, Preparata and Del Giudici, is that these water so clusters of water would go into a quantum state. And to give you an idea of what this could look like, I don't know if you've heard about things like superconductivity or superfluids. And basically what's happening at these like kind of superfluids is at very low temperatures, big ensembles of atoms, like here a little cube of metal, start to have very weird properties as a whole, as a bunch of atoms. Now, in homeopathy, what we think is happening is that it's not so many atoms. Here we're talking, billions of atoms, but in homeopathy we'd be talking about a few thousand molecules at a go, going into these quantum state. And then this is, to give you a picture, the predictions are that these quantum states, these coherence domains, as I'll call them, form about 25% of the volume of water. They would be about the same size as a protein. And then, interestingly, they have the capacity to register the surrounding electromagnetic information. So if you have a protein, proteins are vibrating all the time. They have their own frequencies and sets of frequencies that are essential for their functioning. And in principle, we think that these coherence domains can copy that information. And then there's another interesting aspect of these domains is that the theory predicts that if they have similar information, they tend to attract. And if they have dissimilar information, they tend to dissociate. And then that means that things that have the same information tend to gather together, and those by types of information. And you'll see later that this is one of the reasons why when you have remedies, you tend to succuss. And we'll see how this works in a second. So how does this explain homeopathy at all? I'll show you, the classical picture of dilution is you put a substance, here a protein, it doesn't matter, it could be any substance really. You put it in with water and you start doing your dilutions, but basically after, quite quickly, you've got rid of everything, there's nothing left. And this is what all the scientists say is happening. You've got water, so what happens with now the coherence domains? The thing is a bit different because now we succuss. And when we succuss, what I said about those things clumping together, if they have the same information, the energy that holds these clumps together is not very high. So when you shake, that's enough to kind of separate them out and allow them to spread around your sample. So your information, or your quantum domains which were just around your original sample before are now spreading out and multiplying. So you can see quite quickly how this becomes quite interesting, because now you're getting rid of the original substance, but you're keeping quite a lot of the information. And each time you succuss, you're kind of breaking those clusters apart and multiplying them. And then at the end, you don't have the original substance, but you've got that information you kept from the original substance. So this is, kind of the building blocks of what, we're thinking is happening here. And then it's not far, it's not, the next step is you take those water structures, you put them on the pill, you put them you give them to the patient, and they come back to whatever they were doing in the body and start to interact through the frequencies they have. Okay, so that's, and we know that proteins have a lot of frequencies. For example, all the enzymes have specific working frequencies, and you can accelerate them or decelerate the activity just by playing around with these frequencies. So you can see here how if you think that a domain here might have more like a symphony of frequencies all working at the same time, you could be modulating quite a lot of things in the body at the same time. So that's the basic building block to have in mind when I'm going to present the rest. Now, one of the first consequences of what I just told you is this so-called crosstalk effect. And the idea here is if you have a sample here, like of a homeopathic sample, and here you have a just normal water that has not had any homeopathy, if you leave them side by side long enough, the control, the blank sample, will pick up the information. And this is not just a prediction. This is now seen in experiments. That is, and this is also demonstrated by the fact that if you put some shielding between the two, this effect stops. And this has been seen by Luc Montagnier in his lab using the coils to record information and to analyze information, but other experiments too. So this is the information, the Luc Montagnier experiment I just told you about. He puts his DNA dilutions here, puts it beside a blank sample, and 18 hours later, the two samples register positively on his electromagnetic reading device. And Professor Endler in Austria with the frogs, tadpoles, sees this effect as well. That is, what he did is, instead of giving the remedy to the tadpoles, this is thyroxine, which in this particular experiment, thyroxine 30x slows down the development of the tadpoles. Now here, he didn't put the remedy inside the water, he just put the vial, a clean vial, sealed. And just the fact of putting the vial in the water was enough to see this effect. The effect was working through the glass. A similar effect has been seen by Baumgartner in Switzerland using plants. Basically what he saw is that he had his working plants. He puts them in sheets, so there are like 10 plants like this, and then he would put 10 placebo plants. So 10 plants treated with the verum, 10 plants treated with the placebo. And then after a while, they analyzed the results, and they saw that the placebo plants were not really placebo. They were being affected. And they were affected, the closer they were to the verum group, the more affected they were. And they saw that kind of a linear relationship, like of distance between the plants in the placebo group. The closer they were to the verum, the more affected they were. So again, this idea of action at a distance in some way. So of course, before I move on, this has already huge implications for when you do placebo-controlled trials. I suspect many placebo-controlled trials failed. especially in a veterinary world, where you would just take, the remedy, put the placebo on top, go for coffee, and then you forget that basically your experiment is done. Your placebo is worthless. You just so just a thought, and we can discuss about what are the implications for your practice. There are not many implications for your daily practice, because once the remedies are done, these things clump together, as I said, and they tend to be quite stable in that state. If when you have a blank sample, that tends to register information.
What about liquid remedies stored in a drawer together?
Yes, exactly what I mean. So in the liquid remedies, all these clumps, they tend to gather together, and there they self-reinforce, right? If you have the same information, your remedy is one information or one set of information. So all that stuff tends to clump. And that's stable. It's the blank where there's no single information that is quite happy to take information from outside.
It already has information.
It will tend, because it will gather together and then stay strong. And that's why when you take it out of the drawer, you shake it.
I think it's the same question. I mean, in those where we have all our remedies sampled together,
Yeah.
it seems like they should infect each other.
Yeah, and
Can we?
Okay.
Can we? Maybe we can listen to Alexander's presentation and we take the questions.
I think it's good, because it's probably better to do this.
I'm already worrying about all my test sample, my test capsules, who are lying in big boxes side to side. So I wondered how this new knowledge affects that. Do they turn everybody in the same box all the same, or what is happening?
What we think is happening is that once they're formed, once the information in your sample is unique, that's quite stable. It's when you have a blank sample where there's no specific information there, it's just kind of like a blank page. It just picks up everything that it can from around. There seems to be practice, and because of the way, if they have the same information, they stick together, they would kind of reinforce each other. Yeah, it's the same information. The blank then is just a blank piece of paper, basically. You're not allowed.
Maybe questions will be answered during the speech.
Yeah, maybe I think those are the typical questions, so keep them for afterwards, and then I'll just carry on. So now next is, okay, so I told you water structures. I told you about one effect you should look at for, which is crosstalk, which comes out of theory and is already seen in practice, in like experiments. Now, next one I want to show you is 3 groups in the world, to my knowledge, have been able to concentrate water structures, and you can do it with homeopathy, and you can do it by other techniques. But homeopathy is one of the 3 techniques you can do it. And basically, you make a lot of homeopathic preparations, and then you evaporate using so-called vacuum evaporation the water, and you end up with things like this, which you can see under the microscope or under the electron microscope. And if you make a lot of it, you see something like this. And this is, you can put it into your instruments to measure with the mass spectrometry. This is water, solid, at room temperature. By normal physics, it's impossible. So this is the type of thing that we're working on, because for conventional scientists, this stuff is supposed to be impossible. So very, very strong evidence of that. We're doing something interesting. And again, if you put it into a different microscope, which is called an atomic force microscope, and it's a different microscope because it has a little kind of finger that goes across the sample, you get, again, different images of these aggregates, so these domains I told you about, kind of clumping together in different ways. So here you kind of clouds, and here you see stripes. And these filaments are a very common thing that's seen over and over again in different techniques today. So there's also evidence of clumping. So this is using fluorescence microscopy. They put little beads of fluorescent material, and they see that under certain conditions with these water structures, they tend to aggregate, again in a way that we can't explain with normal physics. And I want to show you some of the work of Professor Pollack in the States, and he's the organizer of the water conference I was at last weekend in Bulgaria, where basically all the water researchers of the world gather, and you have some pretty amazing talks. And what he sees is this. This is what's so-called a naphion layer. A naphion is strongly hydrophilic. It has charges on it, negative charges on it, which means it really likes water. So it's inverse of hydrophobic. And what happens there is quite surprising, is that the water, I'll just start again. The water here forms structures that repel any impurities. And this to quite large extent. They've done it to about 1/10 of a millimeter, which in terms of physics is huge. And so they're using this type of thing now to make filters, to make things. And this surface here, this water that was here, has very weird properties, very similar to the ones we're seeing for homeopathy. We're seeing water structures in a very different, interesting properties. So that's just to show you that it's not just in homeopathy that we're seeing strange water structures. We're seeing them outside homeopathy, coming from different research angles. So in homeopathy, coming back to homeopathy, what we see is some interesting techniques I just showed you, but also we have traditionally had a lot of work with nuclear magnetic resonance. There's been about 30 studies, and I'll show you a bit more a bit later, and that's really interesting. Conductimetry, so measuring the conductivity, so how it conducts electricity, also has been a very rich field and is given some interesting results. And a very promising field is infrared spectroscopy, and that's really coming forward because we're able with this technique to show that homeopathic preparations are different from bulk, from just normal water, and that even different remedies are different. And what's really cool about the infrared technique is that you can analyze the different bands and show that the water structures are actually different in different samples. So it's a really promising technique that we're pursuing. And so when I say pursuing, I'll be pursuing in Heidelberg, where I'm setting up my lab. I just moved to Heidelberg, so in Germany, and that's where I'm setting up all these experiments. So why the fuss with all these water structures, and why should you kind of think about it? Water is extremely odd. We are basically 72% water. We are walking water. Water is at the very heart of life. No protein functions without water. Basic life started in the oceans. We suspect that, in any case, the water structures that we're predicting predate life, and life would have evolved using these structures from the very beginning. And I suspect that what we're seeing is going to be essential to our understanding of health in general. If you think about, all the weird stuff you see in homeopathy, and you think about acupuncture, if you think about, there's a lot of other things that kind of would start to make sense if we look at the body through the angle of water, and how all the water pathways and all that, and the communication that is possible thanks to these kinds of things. So I think there's going to be a vast field of research in the future. So I want to show you work in progress, and it's work we're doing with a collaboration with some people in Switzerland, with Stefan Baumgartner, Sabine Klein, and Sandra Wittenberger, Ursula Wolf. And I'll just show you some preliminary results of the type of thing that we're trying to do here by really going back over all the literature that is in homeopathy about the experiments that have been done, so physical chemistry. So we've got more than 100 publications, and the question really is, what have we learned for more than 100 years of research into the physics of homeopathy? And we really need that for the future. So we had 228 publications. We excluded 73 for different reasons. We got 155 in the end. And just to plot over time, we see that we're publishing more and more experiments on the basic research in homeopathy. And Germany is really publishing the most. France, but actually France, that research is old now actually, because France is not publishing much anymore. Italy is publishing right now a lot, but so recent stuff is coming out of Italy, recent stuff also coming out of India, and so on. So and Sweden. So as I said, electroimpedance, so all these electric conductivity measurements come tops. Analytical methods are all the methods that try and look for traces of things, and nanoparticles and things like that. Those are under these types of techniques. Nuclear magnetic resonance are quite successful, I'll tell you about this. Spectroscopy, so like things like infrared, UV spectroscopy will come under that. And then people have been looking at more odd things like heat effects, or just imaging, or crystallography, things like that here, and so on. So we analyzed all these papers for the quality itself of the paper, because some of them are very old and just don't give us enough information to know what they did actually. So we did that and dismissed the poorest papers, so we end up with about 110 publications. And so on those kind of good papers, we are going to focus now on the ones on NMR. Only 19 were peer-reviewed. 7 of them were really just describing things that had no statistics whatsoever. 25 individual experiments. So one first analysis which is interesting is that the 6 negative experiments were nicely peer-reviewed and of high quality, but the other ones, we've got 19 positive, but only 15 are peer-reviewed, and only 10 are actually good, solid results. So we really need to do more research here to get more, of the good stuff. If you're interested about what kind of remedies people look at, it's mostly very simple remedies: silica, sulfur, histamine, NOx, all the type of typical remedies people look at in these experiments. And then, as I said, one of the big problems is replications of these experiments. It's good that one group does it, but other people need to do it again. And that's a big problem, and we see it here again. People do replications in-house themselves, and they kind of manage to do that. But replications of somebody else's research is very difficult. And actually, in this field, no group that's decided to copy another person's research managed to do that. So we have a real problem. But okay, what kind of things are we getting out of this approach? So one of the things is the conclusions from de Monja, which has been the guy in nuclear magnetic resonance who has been publishing the most, is his conclusions after nearly 20 years of research is that he's seeing something quite consistent, is that the samples, the homeopathic samples, are showing properties that are more like ice. So these are your control samples, these are your homeopathic samples, here 12C, can't remember what he's using, I think it's silica. And he's seeing that, so these characteristic graphs are characteristic of ice type structures. So that's the type of thing he's seeing. So that's one kind of interesting bit that we're getting out of this approach. But I think what's, for me, was really strange and came out of this analysis is something I'm going to call ultra clean, or the role of impurities. And you'll see what's happened. When I analyzed what happens when you use an ultra clean setup, that is, you use highly pure water, that is really strange water, it's super, super clean. And you use very sophisticated glassware that is not going to leach, that is, it won't put the impurities back into the water. So if you use that, you get no positive results. None of the studies that we looked at that used this super, obsessive clean water got results. So maybe we need those impurities. So this is also what Demangeat started to think, and he started to add impurities, to add stuff. So he added salts, normal salt and lithium chloride, and then he got better results. He saw that lithium chloride was the best, and that actually using pure water was the worst. He didn't get very good results with pure water at all. So maybe impurities are actually necessary to the whole process. And you might be thinking, why is this important? Well, it's because scientists, have been thinking it's all, homeopathy is water. So we'll use the purest water we can get. This is like kind of logical thinking, right? But it's throwing the effect out, right? So, now we're talking about this, and we're putting back salts and things that are more like natural and that allow all these water structures to actually form. So this is the type of thing that we're gathering from those 100 years of experience, really. Okay. So yeah, to be able to make better experiments. So briefly, I want to tell you about the research of Stephen Cartwright. I don't know if you've seen what we posted about in Facebook and stuff. It's something that's just been published, and it's the first big piece of research that's come out of the Homeopathy Research Institute. So in terms of publication, so we're really excited. And basically, what's happening here? Stephen is using so-called solvatochromic dyes. Solvatochromic dyes are molecules that have a specific color, especially in water. And the solvatochromic thing means that they have charges here, and that these charges are influenced by the water structure around them, so that the molecule changes color depending on the water structure around it. Okay? So that's a very interesting model, because it, in principle, it can tell us a lot about the water structures I just told you about. So, these are the types. There's a whole family of these molecules. So solvatochromic dyes, and this is what Stephen basically is using. And then they have a property, mostly because these rings are flat structures, and they tend to stack here. So they stack vertically or in different ways, and you might be familiar with this type of stacking with amyloid fibers, Alzheimer's and so on, are similar type of molecular aggregates. So what he sees is that if you put glycerol 50M, then this disrupts the aggregation I just showed you. He's able to show that looking at the spectra of the molecules, he's able to show whether they are more aggregated or less aggregated. And when he puts glycerol 50M, he sees a change in this stacking. It's not only glycerol, also different remedies he tried, but mostly it's been glycerol. And this is typical kind of data he gets. This is for this dye, ET33. And this is the difference for the spectra, difference between the control and when he puts the remedy in. So really it's just the effect of the remedy, and he sees these differences, and he can interpret these differences as a difference in stacking of the molecules. And here, this guy lowers the aggregation of the dye. So the glycerol lowers the aggregation of this particular dye. And this is an effect that peaks at around 100 and 3 hours basically, and then comes down. So you could see over time how this is changing as well. And this is another guy, BDN, and here glycerol enhances the stacking. So we don't understand everything and how all this is happening, but it is happening. And what's interesting is, so as I said, the dyes either enhance or diminish the so the homeopathic dilution enhances or diminishes the aggregation. And what he showed is that the dyes that were most aggregated that get deaggregated, and the dyes that were less aggregated get more aggregated by the glycerol. But we don't understand why, but it's a very interesting finding in the lab, made very, very rigorously by Stephen. So we're pursuing this line of research. Good. And then I wanted to finish by giving you some quick background about the Homeopathy Research Institute, because I'm aware you might not know a lot of the stuff that we're actually doing. So this is our website, Homeopathy Research Institute, hri-research.org. We were created in 2009. We started to fund Stephen 5 years ago. We started Philippa's project a while back. And then we had our first conference in 2013, and we just had our second conference in June in Rome. It was really great. So we're structured that we have a board of trustees, and we have a big advisory board. Now I'm proud to say that it's bigger than this. We have really a lot of really good scientists now on our board. And then we try and keep track of a lot of projects. So, we have 3 fundamental areas. So how does homeopathic medicine work? What can it do? And also databases and things like that. So, for example, Stephen's work is physical chemistry of high dilutions. My lab comes here, Water Research Lab. And we've got Peter Vexpin's project, here, depression. That's Philippa here, ADHD. We've got projects in IBS, bones. And basically, we're quite happy to add more projects for people who want to have more publicity for their projects. We hold a partnership with the Carsten Stiftung in Germany, which are really the world's expert at databases in complementary medicine. And so we basically hold a portal to their database, and so it's a database of clinical trials. So if you want any information about a specific disease, you can type it in there, press go, see what happens. You'll get all the clinical trials for that particular condition. It's the most up-to-date database for clinical trials out there. And also, we've put a lot of favorite answers and questions to all the questions we get from the skeptics. there is no scientific evidence homeopathy works. Scientists say homeopathy is impossible. So we have all these, sentences that people keep asking us, so what do you say to that? What do you say to that? So we've basically, come up with our best answer for all these questions with diagrams, with all the references that you would need to answer back with, the best that we know today. So this is the type of thing that we've been busy doing. We also got clips of various scientists and clinicians. This is Elizabeth Thompson. We are releasing all the films from the 2013 conference. It's going to be free on the web. It should be very, very soon, if it's not already done. So you can catch up on a lot of the researchers, and we're going to release the films from the last conference in Rome as we go along. We're slowly releasing them on the net for free. And if you're really anxious, you can go and buy them already. And then we have a lot of really nice quarterly research newsletters and articles. So each quarter we release a newsletter. The next one is going to be over Robert Matty's review of clinical trials of individualized homeopathy. I highly recommend you read it. It's really, really strong. And it's really important because it's a systematic review that shows homeopathy works, and individualized homeopathy works. So really, this is top stuff. It's just coming up. So the team is Rachel and myself most of the time, and then we have a few people helping us. Simon doing a lot of organizing, Alistair helping us with the communications, and then another consultants and so on. Basically, when we add up all these people, we get to about 3 full-time person. So we need your help, basically. So we need help. We're not, we're trying to duplicate ourselves, but it's proving a bit hard. So that's basically what I wanted to show you. So we need your help. You can make a donation, but I would say it's more about spreading the word. And if you have contacts with wealthy individuals, and we'd like to hear about them. And that's it for me. Thank you very much.
I'm sure there's plenty of questions and there will be time for that. Thank you very much, Alexander. Super interesting as always. The Swedish Association, Svenska Föreningen, our association, is supporting HRI's work, of course. For the ones of you that are not familiar with it, please go into the webpage and spend a lot of time there. It's a jewel.
It's more than 100 pages of information now. So
yeah, so they're doing a fantastic job, and we're very, very pleased.
And the search button on top works, so you can, if it's too big, you can just search for something.
Fantastic. So let's do another one thing I wanted to tell you. All of our speakers have agreed to share their slides with the participants. They will be shared, and they will be you will be allowed to use them for personal use, not for publications or for reproducing speeches yourself, of course, as always. First question.
Hi, Alex. Hi. Great to see you in Sweden again.
Thank you.
And next time you set up a research project, why not do it in Sweden? I mean, you've been to France and now in Heidelberg, and next time here. I have a question.
I just answer that point. If I may. We were talking about this with Yale. Doing research in Sweden, great idea. But I want to stress, and this is something for all of us, is that what we're really missing is big trials. We've done a lot of research with small trials, and they consistently fail to convince the health authorities, mostly just because they're small. So we need to start thinking about Europe-wide collaborations, clinical collaborations, where we can get 500 patients together, maybe over different centers. So this is the type of thinking that we're working.
That will be great. We're in anyway. Right. I have a question, and it was when you talked about the crosstalk. And let's say you have different mother tinctures in the same room. How far apart would you say that you would have to place them and not to have a crosstalk?
Well, we know that from experience with anybody who's worked in a pharmacy will know that, thank God, this is not a problem, because otherwise none of this would work. So mother tinctures and all that don't seem to be affected by proximity, because we know they all stack together in the pharmacies and they work. It's really your blank samples that you need to think about. But for most of you, this is not going to be an issue, because it's only if you do trials, the placebo-controlled trials, where you need to be very careful. Because that's where, you need to be sure that your blank sample is really blank. And that's what you need to do.
If I may ask another question, when it comes to combined remedies, that what you're saying is that the remedies or the substance that look alike, they climb, they get together, and if they don't look the same, they don't mix. So,
well, yeah, very good question. And if you ask the pharmacists, they'll tell you they have interesting ways of making combinations. That is, they don't just mix two homeopathic remedies and sell it to you. They usually mix them and potentize that before giving off the... So, you see what I mean? It's like they potentize the mixture, and that seems to work. But the mixture itself, I don't know.
Thank you.
So what about keeping the unpotentized liquids like a couple of feet from all the potentized remedies? Is that enough room?
We don't know. I mean, actually, if you have many, like in the pharmacies again, the water they use, or the 70% ethanol they use for making the remedies, is sitting in the middle of the room usually with thousands of the remedies just beaming out information. But I think it's the same thing as if you were in a room full of people talking to you, you can't hear anything. If there's too much stuff happening, if there's too many remedies around, what is he going to do? Nothing happens because there's too much happening. Does that make sense? It needs like one thing to be able to capture it. Like there's an experiment where they did a proving, and if there's just two remedies and a blank, the blank doesn't know what to do. Okay? So just two, so it's like two people talking to you. You can't, it's just, you need one person. And then it can latch on. One type of information, then it can copy it. If there's too many, it just doesn't manage.
So you're basically saying you need to put the blanks in a different room.
Well, I'm saying you don't need to worry about your blanks, because they're not, if you probably have many remedies in that room.
Yeah.
So the blanks, they just don't know, they're not going to copy anybody.
Oh, I see.
So because too much happening. If they were in a room with just one remedy, then I would worry.
Oh, I see.
Yeah. And this is, you do this all the time. When you give a patient a remedy and you say just put it in your pocket or put it by your bedside, that's what you're doing basically, right? You just say just carry it with you. Basically, you're doing this crosstalk effect.
Yeah, apparently this crosstalk effect is is fascinating.
Yeah.
And there's a lot of questions coming up. One little bit curious or question is: do you consider a homeopathic remedy which has found its form more stable than a human being?
Sorry, say that again. There you go.
Do you consider a homeopathic remedy which has found its form more stable than a human being? And I will have a question after that.
Well, okay, next.
Okay. Because what I'm thinking about is that, all this about intentional homeopathy also.
Yeah.
So in clinical randomized double trials, we will have a big problem. And one of the problems is that if my intention, if I have a patient and it's a clear sulfur, and the sulfur patient is in the placebo group, my frequencies, and if I have the placebo, and I'm going to give the placebo, we have a big problem.
So, okay, very good question. Really on the ball. Okay, yes, it's a big problem. That's why it should be a pharmacist who gives the placebo, first thing. Second thing is the intention. Yeah, we suspect their intention can play a role here. There's experiments by Emoto, by other people, indicating that intention could play a role. Now, what I'm really interested in is if you do the placebo randomized controlled trial, so you do it right, and you just look at what's in the pill, that's the effect. Because, of course, if you start talking about intentionality, you're going into placebo effects, blah, blah, blah. It's getting very complicated. So this is why, people don't like the placebo randomized controlled trials, but actually, for this particular question, they're really quite interesting because all problems of placebo are resolved, and you get still an effect. And the review coming out from Robert Matty is about this: is if you do individualized homeopathy using randomized controlled trials, what do you get? And you do get positive results, very positive results. So it does work, and it tells us that the remedy itself carries information. And that's beyond any intentionality and so on. So that water is able to capture whatever the homeopathic remedies are carrying information.
And the answer on my first question.
More stable. I think that goes back, for me, to a very long-term question of these structures are, it seems, quite essential for life. And it's quite tempting to think that life would have evolved around these structures and using these structures in different ways, as information, as ways of communicating. So, it's quite tempting to think that we are a complex water. This is now dreaming here, but that we are water made solid by the cells and so on. And what if we were water networks, very subtle water networks, that the matter that we have is just kind of keeping together, right? And that the most fundamental of us would really be water. Because I can go into the physics of it. Water has extremely interesting properties that are really weird. And I can't go into the details, but it has very interesting properties that make it much more interesting than a lot of other. It's very sensitive, very sensitive. So anyway, yeah.
So we have time for the one last question.
I'm wondering about the process of making homeopathic remedies. To transform them from the liquid to the pills. Then also alcohol is added. So what happens to the structure of the water?
Okay, I'll answer the alcohol first. It's funny because I was discussing this with the researchers last weekend. Alcohol, they saw that 10% alcohol actually is the best. So probably the old experiments with using brandy and actually that's the best amount of alcohol. Now 70%, there's still 30% of water. That seems to be fine. So there's no problem there. Sorry, your first question was pills. This is the most difficult aspect we have now. However, it's recent experiments using the water structures I showed you, the ones you can actually see. These guys are extremely stable. That is, they're stable up to 600 degrees Celsius. They resist boiling, right? So they will completely resist being on a pill. They will be there. So they go on the pill, the rest of the water evaporates, the alcohol evaporates, no problem. And then you take the pill, and then they just rehydrate. So there's no problem about the water structures reintegrating water-based system and then being okay. They're very, very stable, it seems. So.
Thank you.
Fantastic. Thank you so very much, Alexander.